Bismillahirrahmanirrahim. Assalamu alaikum and a very warm welcome to our international audience. Today we have got a very specific topic about inherited retinal diseases. Inherited retinal diseases were thought uh, untreatable diseases and there was no solution earlier. But with the passage of time, with the advancements in gene therapy, new hopes are emerging and new ways to treat are coming to the light. So today we are having our international speaker, Professor Bikash Patnaik. Professor Bikash Patnaik is a clinical director, visual electrophysiology at the University of Wisconsin Health System, Daniel Albert Chair of McPherson Eye Research Institute. He has got extensive experience in gene therapy and especially particularly talking about the inherited retinal diseases. He has also worked on ion channels pathy and abnormalities in ion, particularly potassium and chloride channels, abnormalities which lead to Leber's congenital amaurosis and Best disease. We are very fortunate that a scholar of his repute, experience, and knowledge has given time to educate us about the recent advances in gene therapy in ophthalmology. So, without further delay, I will request Professor Bikash to please present his talk.
Yeah, so sorry for the confusion, but yeah, thank you for having me. It's a great pleasure to share with you all the advances in gene therapy for retinal diseases, and more specifically to talk about Leber's congenital amaurosis and some of the channelopathies that we study in the lab. So I thought of introducing briefly about what this is. Like for example, this title has central dogma provides the framework for molecular medicine. So central dogma in any field of medicine or biology starts with DNA to RNA to protein. That's how we get. So gene is basically your DNA, and then it gets transcribed to RNA, and then RNA gets translated to protein, which is the functional unit in the cell to give us what we are, right? So I just wanted to start with that. So if you see here, this is a brief intro about how a gene. So can you hear the video, the audio?
Yes, Prof, we can hear you.
So
No, Prof, we cannot hear the video. We can just hear your talk.
Oh, you cannot hear the video? Okay, that's fine. So, so as you see here, so what this represents is this represents a, let's say, a gene. So a typical gene will have a start codon, okay, which is a DNA sequence, and then this is the whole sequence. Then you go to a stop codon where the gene frame works, ends, and this basically gets translated to a protein. So the way this works in the cellular system is these are transcribed or these are translated by ribosomes, which are in the cytoplasm of all the cells. So if I show you that this robot that you see is a ribosome, then the robot will start at the start codon. If you see here, I'm showing. So the robot starts here at the start, and then it will go through the sequence, the whole gene sequence from start, so till it ends it at the stop codon where the gene synthesis will end, right? But in certain conditions, what happens in this gene, there are point mutations, and then you introduce a stop codon in between the gene. So it doesn't see the normal start codon, stop codon, but it sees a stop codon before, much before the actual stop codon. So if I use the ribosome now, it goes and then stops halfway through, or it doesn't make the full protein here. So that's a big problem for many diseases. And the way this can be addressed, or we thought this can be smartly addressed, is somehow we can bypass this stop codon so that the ribosome goes from the start, somehow skips or jumps, and then gets to the end so that you get the full length at least recognized by the cells. You see here. So what I have done is I have somehow using drugs obscured the premature stop codon so that now I can get a normal protein. And that's basically the mechanism that we have been exploring for our gene therapy. So what it is here to show you some examples. So this is your, let's say this is the gene here. If you can see my pointer.
Yes, Prof, we can see.
Okay, so this is the gene. It has a start codon, and then one code is here is UGG, and then you have a stop codon. So typically, this UGG is recognized by a tRNA which carries a tryptophan in the sequence. But if this UGG gets converted to a UAG, so it's just one nucleotide chain G to A transition, then it becomes a stop codon. So this tryptophan tRNA will not recognize, normally not recognize. And similarly, we have only few stop codons, basically three stop codons, UGA, UAG, and UAA in the biological system. So what we thought is if we can manipulate this UGA, UAG, and UAA, so we can probably treat all the stop codon related diseases that we encounter. And that's exactly the mechanism that we have been of late developing. So to give me a second, it's not advancing. Okay. Okay. So let me do it here. Okay. So if you look at cell here, so a typical cell that they'll have the membrane, and in the membrane there is one type of protein which are called channels, ion channels. The function of these ion channels is to carry ions like messengers. So I can talk to you, I'm speaking, you can hear me. So that's a communication, way of communication. So the cells, they communicate to each other by passing these ions. So one ion will move from one cell to the other cell will realize something has happened to the other cell, so let me communicate or let me start reacting to that. So what happens in certain diseases or in channelopathies, these ion channel proteins, they get mutated, so they become non-functional. So when they are non-functional, these ions, they don't move, so cells basically naturally realize that something is wrong in the system, something is not working. So, so there are physical principles by which we can define this. Basically, if I take a electrical circuit, so we all know about voltage. We see in America it is 110 volt. If you go to Europe, it's 200 volt. So the volt is basically defined by how much ions are moving in the circuit. Okay, so what is B is directly proportional to I, provided we provide them some resistance. So what are resistance? Like for example, if I use a fan in the room, that's a resistance. If I use a bulb in the room, that's a resistance. So in the cellular system also, the same principle is used by the cells. So when an ion is moving from the outside to the inside, it generates a current, and then that derives the voltage across the membrane of the cell. Like we say that the neurons are firing, and because they are at a certain voltages. So that's exactly how it happens. So these ion channels, they play with the membrane's voltage so as to communicate. So what we did is because, so now we know that ion channels are important for our health. They are almost present in all cell types. And then they are, when they are non-functional, they cause diseases such as vision. So those who are facing vision loss due to channelopathies, they deserve a hopeful future. We should do something for them so that they are able to see. But the problem is, these are not attractive for pharmaceutical companies because we have only a handful of patients with these conditions. So that's our goal, that, how to treat these patients who are n equal to 1 or very few patients in the world. They can be treated by one drug which will target multiple of the genes based on one condition or one mutation type. That's the goal that we are working with. So towards that goal, so what we use in our lab is we use multiple disease models. So one of them is a stem cell. So a stem cell is, if we take a patient, I mean, we can take any biopsy samples from the patient, may it be skin, may it be blood, may it be, I mean, other urine samples from the patient. And then using that, we can make their cells into culture, and then we can program these cells so that we can make the cell type of interest. So in this case, in our case, we are interested in the eye cells, or we can make cells which represent the eye of the human being. And in that case, because the patient has the mutation or the patient has the disease, so those disease will also be manifested in the cells that we get from the patient. So that becomes easier because we don't have to bring the patient to the lab. So we can study the patient in the lab using these cells. So that gives us some mechanism to prove that our disease mechanism that we are proposing is correct, and then also the therapy that we are developing works out. Then the other thing is, how do we compare that? So to compare that, we also generate some mouse models where we introduce this disease that we see in the human, and then they manifest some clinical features that we see in the human being. For example, here, if you see in these mice, so in the control on the top, so if in the control on the top, these are clinical measurements of eye retina OCT, where we know the retina thickness or the integrity of the cells. And then also ERG, electroretinogram, where you see this is the photoreceptor response by going downward, then the bipolar cell response. So we know that the retina is functioning normally. And then this one is a C wave, which represents the function of the RP cell. So in a control mice, we see all of them. That's normal. The structure is normal. Function is normal. Retina is functioning normally. Whereas in the knockout, when we knocked out this particular gene which interests us or which causes the disease, then you see there is reduced A wave, B wave. The structure is altered because there is thinning of the retina, and then also this complete elimination of the C wave. So this gives us some characteristic for seeing that, okay, we are getting the disease in the animal model or in the human cells, and then when we treat, what do we get? So with that in goal, so now I wanted to introduce the two diseases that we study, which are of importance. So one is Leber's congenital amaurosis 16, and then the other one is BEST. I mean, mostly to Middle East people, I will say that, okay, it is more prevalent. LCA is more prevalent in Middle East than anywhere else that we have seen. And then same also with BEST. That's why I thought we should, I mean, this is pretty appropriate to address these two diseases, how we are dealing with it in the laboratory stage, or how we are bringing it to the clinic through our approaches. So LCA16 is an autosomal recessive disease. So as you see here, I present you a case of a tryptophan 2A stop codon mutation at position 53. And then this is an early onset disease where you see there are retinopathy pigmentary changes in the retina. And then you also have other genetic conditions like nystagmus, strabismus, cataract, and then also reduced ERG. And then the visual acuity is pretty poor, 2200 to 2400 in this patient. And then if you compare that to BEST, BEST can be autosomal dominant or autosomal recessive. So here we are presenting one of the recessive cases where there is also a nonsense mutation, but it is an arginine to a stop codon. And then with the BEST, you have these vitelliform lesions. There is abnormal OCT. EOG is abnormal. The Arden ratio in the EOG is less than 2. And then the patients, they have low visual acuity. So if I show you examples of this, so for example, here, these are the channels that I talked about. So this is a potassium channel in the retina pigment epithelium cells, which causes this disease. So in the normal individual, this black trace, if you see, it makes a inward bend channel where if you change the voltage in this axis or the cell, then it represents or it responds with a flow of ions, which gives us this current. But in the disease cell, which is in red, it's basically a straight line, and then you don't see the signature current here. And if you see here, these are the RP cells from the patient where you don't see, so in the normal individual on top, you see this presence of the channels, whereas in the disease cell, they are not present, they are absent. Same thing also for the best, although it is not a potassium channel, it is a chloride channel. So here, if you see, these are the normal patients where you have the best one is present, whereas in the diseased cells, the best protein is absent, and then also the current here in red is also flat. So it doesn't have any current. So that gives us a visual representation of what's going on in this disease. So why this is important? Because if you look at inherited retinal degeneration, there are more than 300 genes and variants that cause disease. So, and then most prevalent is ABCA4, OS2A, and these are very hard to treat because of the size of the protein. Those are large. Whereas, the ones that we study, they are here about 1% or 2% of the total IRD cases. So that's why we thought that we will do a transgene approach so that the therapy that we develop, it will be applicable for all. So everybody is interested in that. So here, for example, I'm showing you the Best disease, which is very low in prevalence, and there are several mutations that affect the protein. Same thing with the LCA16, it's because of the KIR 7.1. There are several mutations we have studied, and then also others have reported that cause this disease. So what it is here, this is the RP cell. So the RP cells are present very posterior in the eye, beyond the photoreceptors, and they basically help the photoreceptor, which are the photosensitive cells of the retina, to function normally. And if these RP cells are not functioning normally, then the photoreceptor also see that, okay, there is a disease, and then they also don't respond to light. And that's how these diseases they cause. So, our approach, as I said, is DNA is present in the nucleus, and then they are transcribed to mRNA. So mRNA now exits the nucleus, and then they are translated to protein in the cytoplasm or by the endoplasmic reticulum. So if you have a normal mRNA, not mutated, then they'll make a normal protein. But if you have a premature termination codon or PTC, then they make truncated protein, which is not a full length protein, so they don't make a channel. So what we thought is we can target this by just not doing anything although we have developed gene therapy, which is mostly targeting to the nucleus. So what our thought was, let's not target the nucleus because, we know that some of the gene therapy, they have deleterious or they cause cancer because they are affecting other genes. So let's not affect anything here, but then let's try to treat this disease here in the cytoplasm without changing the genetic makeup of the cell itself. So how? Just by targeting this premature termination codon. So these are our collaborators. We have several collaborators. So the way this works is, okay, I wanted to also introduce you. The way you study these ion channels is by a process called electrophysiology, where you can see on the right here, this is a normal manual electrophysiology device where you identify these cells here in the screen. So like, for example, these are individual cells, and then I can reach out to each cell by a pipette, you see here, and then we can measure their electrical activity throughout time, how these cells respond to normal blockers or activators. But then this is very time consuming. We are going one cell after other, and then sometimes the cells will not work. So what we have done is we have sped up the drug discovery process by using a robotic electrophysiology system. So where it does is, as you see, this is all automated. So the robot will pick up the cells and it will drop to these chambers. And in one experiment, we will get results from 48 different cells. So we are speeding up the discovery of the drug or treatment. So here you see one example. So these are diseased here. So almost you see flat line here. Whereas after the treatment, you can see some of the cells, not all of the cells, some of the cells or most of the cells, they start to show you a current. And when we compared here, for example, this is the wild type distribution, and this is the mutant or the disease cells, which has been treated with the drug that we have developed. In this case, it is a genome editing. So basically targeting the genome, which to edit the particular nucleotide. So by editing the nucleotide, you can see the treated cells are no different from the wild type cells. So effectively, we have treated a disease here. I'm showing comparison of 65 cells, wild type 65 cells, or healthy 65 disease, and there is no difference between the two. But then this works at the cell level. Does it also work at the animal level? Because I said that we also use mouse to study this. So here is an example. So this is a mouse with a normal response, but then if we knock out, or if we treat, or if we disease the mouse, then we see a decrease in their response, and then you see cell death here. So retina structure is damaged, the electric physiology response is reduced, and everything. But once we treat here on the lower case, so see, now the retina structure remains, it doesn't get affected by the treatment, and then also we see a rescue in the electrophysiology response. So practically in mice, we have shown that what we did in the cell is also applicable to the animals, so that this can easily be delivered to the human eye to treat these conditions. So these are some examples. So again, for the BEST disease also, we did the same thing. So, okay, we can treat one disease. How about BEST? Because that's a different protein. Does it also respond the same way? So here, for example, you see, this is a comparison of the mutant. This is a diseased one in the red. And then after treatment, you start to see they are responding as you will see for the normal cells. So both in case of the LCA and then BEST, it worked. So how about this therapy for the PTC? So here is an example. So these are the healthy here on the left. These are diseased where you don't see the protein is expressed. And this is after treatment for the PTC. So you start to see the protein. Not only you start to see the protein, but you see the protein here, which is normally functioning in terms of the cell physiology. So we did again the same approach. Does it also work, all this nonsense mutation treatment, does it also work in mouse? So here we are showing where we delivered it by showing some reporter present in the mouse eye. And then here we show that if this is normal, this is diseased in yellow, in green is our treated one. So we first cause the disease and then we treat. So progressively we see that the cells are responding, or the animal is responding to normal vision. And then these are again for the BEST. We did the same thing. So these in black is the diseased ones. And then here there are multiple therapy. This is the normal healthy one. So some therapy, they work almost effectively so that you get to the same level as the normal channel function. So what I showed you, so I showed you some examples where we have taken the patient cells. This was the first time that we took patient cells to study in the lab and then compare them to in vivo, or compare them to actual animal studies where they go hand in hand, because not only in terms of the disease, how we see them, but also in terms of the therapy. And through this, what we have done is we have sped up, or we speed up the therapeutic process, where I showed in last 10 years, we have shown that, okay, we can treat these diseases which are rare and ultra rare by gene editing, gene therapy, sTRNA, and then also read through drugs. For example, if you go to a clinic, and then, if a patient walks to the clinic and then the patient has been diagnosed with a genetic disorder, so we don't have an option for them. All we say to the patient is, okay, so you can come back and then let us do some studies and then do it. Whereas when we go to a restaurant, we see a menu card and then we have all these options. I can go for the appetizers, I can go for the main menu, I can go for the dessert, whatever I want. Why can't we offer that to the patients? And that's exactly our goal. So here, for example, you see for a rare and ultra rare disease, I can give them a menu card. I have all these options for you. This will work under this condition. This will, if you are too hungry, I'll have you go for a main menu, so where you can fill your stomach. If you are not that hungry, you want to just taste something, you can go for appetizers. So similarly here, I can give them a customized drug therapy for their specific conditions. And this is, this has been a massive effort, and this is surely a collaborative study to address health equity for ultra rare conditions, which are generally not attended by big pharmaceutical companies or big companies. So I'd like to thank a lot of the people, those who have contributed to this. Of course, our collaborators here in RED, but also the lab members who do the hard work. We have scientists, we have doctors who are working in the lab to address all this so that we can bring something or we can bring hope to the patients that we see in the clinic. I'm happy to take questions.
Well, thank you very much, Prof. You really explained this topic, which is very technical, comprehensively, and very easy to understand for the clinicians also. Just a few things I want to ask you, Prof. Uh, from the patient perspective, if a patient is coming to the clinic for examination and he has got these retinal degenerations, and doctor tells him he has Leber's or these things, how should they proceed, Prof? In the
Right.
management.
So yeah, so this we get we get asked in the clinic all the time. So a patient walks into the clinic and then the patient is diagnosed with certain conditions. Basically what the clinician will do is they'll examine the eye and then tell the patient that, okay, I think you have this, but then there are other tests to confirm that this is. So at that time, if for a patient or for a family, they are already devastated because they know that this is a health disparity. This is a condition which is going to stay there for the life. What next, right? That's a big question that the patient has. So when the patient has a question of what next, how are we advising them as patients? Are we advising them correctly? Like, for example, yes, we will say that you can come back for gene testing, and then the option for the patient is either the patient complies or the patient doesn't. I would say that for the patient, they should comply, because when a doctor says that, okay, I think that this is the condition or this is the disease we have, it is based on what the doctor is observing, but not necessarily it's a final clinical diagnosis, because all the tests which will confirm the disease has not been done. So some patients, what they do is they don't even show up because they are already devastated. They don't even come back. They don't even consult, or they can go to a second doctor, third doctor, fourth doctor, five doctor. The entire life is gone before we realize that, oh, if we had known what exactly the condition, we could have treated this patient when the patient was a year old, compared to now the patient is 10-year old and we don't have a choice because the cells are not there to treat, right? So that's why my advice to the patients and then families, yes, it is devastating. In fact, for a doctor also to give this message to the family, they have to think about it. They have to certainly confirm that, yes, this is the correct diagnosis that I am doing. So with a lot of thoughts, the patient has told you or the patient has given the verdict, so as to say. So take it. Yeah, it is sometimes difficult, but then take your time, go back to the clinic or consult the clinic. You can call the counselors and then talk to them about, okay, so this is what we got, what are our next step. So what I meant by that is we want the families to be curious. We want the families to be properly educated about what the next steps are.
Right, Prof. Prof, you're talking about these channels, Leber's congenital amaurosis and Best disease. Prof, what about the drug delivery? About particularly talking about your research, what will be the
Exactly.
delivery method, Prof?
Correct. So it all depends on what the drug is, right? Like, it's almost like if I am delivering a gene therapy product. So basically, gene is a biological molecule. So how do we deliver a biological molecule? So we cannot deliver a biological molecule in capsules. So to deliver that, basically the capsules for a biological molecule is a virus. Okay, because we all know that we get flu, we get COVID, we get all this because those are all viral. So a virus will come and then, transduce the cells of our body, affect the immune system, and then we start to react. So same way, what we do is to deliver a biological molecule or biological drug, we use virus, but then these viruses, not to, bring fear in your mind. These viruses are what you call engineered so that they don't have any infectability. They don't create any inflammation. They don't create anything. All they do is they will go sit on the cells, deliver the biological material that they are carrying. basically as a carrier or as a courier. They bring it to the cells, deliver it, and then they are done. So they don't, proliferate in the cells. So the problem is when the virus proliferates, then we have a problem because it will go to other part of the body. The other thing also we don't have to worry about is because if you look at the eye, it's pretty confined. I mean, there is a barrier between the eye and then the blood, eye and then the bronch. So anything that you deliver to the eye generally doesn't get crossed to the blood supply so that it will have a systemic problem. So that's why eye is a safe compartment where these drugs can be delivered. So if it is, as I said, if it is a biological molecule, it can be delivered by virus. But if it is not a biological molecule, then you can have other options to deliver it. Like, for example, you can have lipid nanoparticles, which are basically again cell-like. So they will have it's almost like a micro bubble which carries these molecules, smaller molecules. Then they can also go. You can make it very specific so that it goes to only one cell type. It doesn't go to all cell type, because every cell will have some specific receptor or some signature receptors on the membrane. So these will go sit on that particular cell type and release the material to that cell so that the other cells are not affected. And in sometimes what happens is we worry that, okay, if we deliver and accidentally it gets to another cell type, what happens? So you are muted. Yeah. Yeah. So even if you deliver it to the other cells, also what happens is because the gene only expresses this one cell type, it generally doesn't affect the other cells.
Thank you, Prof. It was, in fact, very informative talk, and really you came to the levels of the clinicians to explain this very challenging topic of gene therapy. And we know this is the future of the medicine. Gene
Absolutely.
therapy, the future lies over there. The journey started with the antibiotics and all these things. I believe perhaps now we are entering into this future. So I want to thank you once again on the behalf of our audience. And I know you are a very busy person, but you still gave time. I'm thankful to you for this. And we're looking forward to more collaborations with you in future. Thank you, Prof.
Absolutely. Thank you. Thank you.